right, well, let's get started. It is 4 o'clock, and I just want to welcome everybody to this CaTS seminar series, and this continues to be a great success. Today, we're hosting Tulane, we're hosting CaTS, and I wanted to also take this opportunity to thank everybody for their participation in the series today. And since this will be the last one before the end of the year, I want to wish you all a happy holidays and happy new year, and look forward to seeing you back here in January. So with that, I'm going to hand off to Dr. Bojanowski, I got it, and Dr. Kolls. Christina.
Thank you. So it is my absolute pleasure to introduce Dr. J. Kolls. Dr. J.K. Kolls is the John W. Deming Chair in Internal Medicine. He is a professor of medicine and pediatrics and the director of the Center for Translational Research in Infection and Inflammation at the Tulane University School of Medicine. He earned his medical degree from the University of Maryland. He completed his internal medicine and pediatrics residency training at Charity Hospital in New Orleans, followed by fellowships in adult and pediatric pulmonology at LSU and Tulane Health Science Centers, respectively. He completed a research fellowship in the laboratory of Dr. Bruce Beutler, who was a Nobel laureate in 2011, at the Howard Hughes Medical Institute at UT Southwestern Medical Center in Dallas, Texas. He was recruited in July 2003 to the Children's Hospital of Pittsburgh at the University of Pittsburgh in Pittsburgh, Pennsylvania, where he was Division Chief of Pediatric Pulmonary Medicine and the director of the Richard King Mellon Foundation Institute for Pediatric Research, also at the Children's Hospital of Pittsburgh, UPMC. He joined Tulane faculty in 2017. Dr. Kolls has been a member of the American Thoracic Society and is a very well-respected member of the society and a leader. He has been a member of the Allergy, Immunology, and Inflammation Assembly and is also currently serving as the chair. He has served previously as the AII Program Committee chair as well, and he has also chaired the Gordon Conference on the Biology of Acute Respiratory Infection. He has also organized a Keystone Symposia on Th17 cells and asthma and COPD. He is a member of not only ATS, but also the American Association of Immunology, the American Society of Microbiology, the American Society of Clinical Investigation, and the Association of American Physicians. He has co-authored or authored more than 300 peer-reviewed articles. He has been continuously funded by the NIH for 25 years for his important work on lung and mucosal immunology. He is, honestly, I'm getting like flustered because Jay, this goes on and on. I can keep on talking about you. So I think it's just fantastic. So, and he is my mentor and I just respect him dreadfully. So I'm very happy to present him to everyone here today. Dr. Kolls will be presenting his work on targeting ACE2 in COVID and also sharing with us some thoughts on GWAS studies. Dr.
Great.
Kolls.
Thanks, Kristen. My screen here. Can everybody see that? Okay, good. Yeah, so well, first disclosures, I'm used to giving CME talks. So I'm a consultant for Boehringer Ingelheim on severe asthma, but of course we haven't met the last year because of COVID. And a consultant for PH Pharma in a drug that they have for cystic fibrosis, a neutrophil elastase inhibitor, which won't be the subject of the talk today. So my work has been supported by NIH, and we've completed a grant with Novartis that is not related to today. Well, the one disclosure I have is the molecule I'll talk about is that Tulane has filed a provisional patent on as a potential COVID-19 therapeutic. So the learning objectives today is to briefly review COVID-19 pathogenesis, including genetic risk factors that have been identified to date, review and discuss preclinical models of the disease that inform, insight and pathogenesis, but also how these models can also help inform vaccination strategies as well as therapeutic strategies for COVID-19. So, prior to COVID, had a longstanding interest in pneumonia, having trained with Steve Nelson and Judge Shelato at LSU. And shortly after my fellowship, pneumonia surpassed diarrheal disease as the #1 killer of children in the world, and particularly children less than 5 years of age. And even in the United States, it's the #1 cause of child absence from school, and also missed parental missed days of work. So respiratory infection, not necessarily pneumonia, but respiratory infection. So even in the U.S., it has a significant morbidity. So when COVID hit, it actually we were already kind of working on lung infection, and we had a lot of genetic tools to apply to COVID. So this is a representative radiograph that was published in the British Medical Journal a few months ago. COVID with these kind of patchy ground glass bilateral infiltrates. I'm sure Christine probably has a number of chest radiographs that she could share with you, different stages of the disease. But it's typically a bilateral disease as opposed to like a bacterial pneumonia, which is sometimes just lobar disease. And the pathogenesis is complex, but we think that in mild infection, there's emerging evidence that in mild infection, for some reason, at least one etiology of mild infection, it could be a low inoculum, but the other thing is there's data that suggests that patients that make a T cell response faster or more efficiently seem to have milder disease, as opposed to antibody responses, which can sometimes be linked for severe infection. So it's oftentimes you can find anti-SARS-CoV-2 antibodies in patients that are still have pretty much florid ARDS. So it's thought that there's many target cells that get infected. As I said, the initial entry receptor is ACE2, and ACE2 is expressed on the nasal epithelium all the way down to the lower respiratory tract, including type 1 and type 2 pneumocytes. It's thought that these type 1, type 2 cells get infected, which may create some barrier dysfunction, clearly surfactant dysfunction, leading to ARDS. There's some debate in the field of what this extent of endothelial infection is with this virus, but it's thought that part of the late stage severe COVID is an inflammatory reaction with an influx of neutrophils and macrophages, and toward that end, some anti-inflammatory strategies like dexamethasone, and a recent study with a Janus kinase inhibitor appears to have promise in terms of actually improving survival. There is other organs are affected, particularly there's myocardial dysfunction that occurs, there's hepatic dysfunction, there's renal dysfunction, and how much of this is due to the systemic inflammatory response versus viral infection, it remains unclear, but at least the studies that have looked at viral RNA and autopsy specimens have largely found the viral RNA confined to the lung, with very little evidence that there's significant viral RNA in distal organs. So this may be a response to the inflammatory response. So as I mentioned, SARS-CoV-2 binds to ACE2, but there is an enzymatic step, and one of the proteases that clips the spike protein that's on the surface of the virus is TRM Cells. But during an inflamed lung, there's going to be another, it's going to be neutrophil elastases, there's going to be matrix metalloproteinases, and the relative contribution of all these proteases and how, in terms of progression of COVID, I think remains to be determined. There's thought to be, since ACE2's host function is to regulate angiotensin II cleavage, that there's some theory that perhaps the virus downregulates ACE2 expression and reduces angiotensin II processing, which contributes to some of the hypercoagulability and microangiopathy that's seen in the organs of these patients. And that this clotting hypercoagulability can obviously lead to more distal organ failure, such as cardiac disease and kidney failure. So this is one of the first autopsy studies that showed basically a vascular endothelialitis and thrombosis and this microangiopathy that's been described in the lung, and microthrombi in the lung. And then this is EM pictures from the same study, and you can see that there's this is a healthy capillary endothelial bed in the lung, and this is obviously here you can see this is grossly disrupted here. So there's a lot of endothelial injury and microangiopathy in this disease. This is an autopsy study actually published by Dr. Vanderheid's group at LSU in an African American cohort, and you can see that he also found evidence of microthrombi and microangiopathy in these severe patients. And you can see that there's this just diffuse alveolar damage phenotype here, and you can see these very thickened fibrin-like membranes here in the distal lung, which obviously is going to impair gas exchange across that surface. So this is a study that was published in JAMA Internal Medicine, looking at comorbidities and other risk factors, and you can see that age is a huge. There's almost a dose-dependent effect of age here, with mortality even increasing in people in their 40s. Male sex is a consistent finding across our most recent meta-analysis of over 3 million COVID cases, which is pretty impressive data mining to accumulate 3 million COVID cases around the world. So that this is, there's something going on with the male sex that that is across many countries that would argue that maybe it's just not diet or other factors, that there's something about the I'll when we get to the end of the talk, when I talk about the animal models, we do have one animal model that definitely shows a sex difference where the male mice do worse than the female mice. And then BMI is a risk factor. Of course, diabetes, anything that suppresses your immune system, like active cancer, is a risk factor. So these are in the ICU or the PI2 FI2 ratio. So basically, this is a measure of how much oxygen one needs to provide to get a PaO2. So the lower the number, the worse the oxygen transport is in the lung. And you can see that mortality goes up significantly, again, in a dose-dependent fashion. So ARDS is typically defined as a PaO2 FI2 ratio of less, severe ARDS is less than 200. Again, the SOFA scores, that's a severity of illness score used in the ICU, and that trends with mortality as well. So the interesting thing is children seem to be immune to this disease, and which is kind of the opposite of the 1918 influenza pandemic, where young adults were a significant proportion of the people that succumbed to the H1N1 strain from 1918. And this is the current thinking of why this is not as common in children. One is that there's less comorbidities. There's been hypothesis that there's differences in ACE2 expression. I think the jury's still out on this one. Actually, ACE2, when you look at nasal epithelium, ACE2 is higher in children than it is in adults. This may support the kind of the nasal shedding that has been described in children. There's potentially a role for thymic function, and actually we have a faculty member here at Tulane, Derek Pochask, who is actually studying this in the context of high path H1N1 influenza, where the thymic function clearly seems to regulate the severity of influenza pneumonia. And Derek's working hypothesis is that maybe these T regulatory cells, which are important and derived in the thymus, but control autoimmunity, also may be controlling the severity of inflammation in severe influenza. And I think the most interesting hypothesis too is this cross-reactive immunity. So I'll show some data along these lines. So there's a number of alpha and beta coronaviruses that cause common colds in children, and some of these have genetic homology with SARS-CoV-2. First, the ACE2 gradient. So this is a paper published by Ralph Barrick's group at UNC Chapel Hill, and some people may know Rick Boucher's name. He's world-class expert in ion transport and epithelium, has worked on the cystic fibrosis gene for a number of years. And then Scott Randell, who his lab actually cultures primary human epithelial cells, both from the lung and from the nose, on what are called these air-liquid interface cultures. So these are nasal brushes, and then they're put on transwell filters here, where the filter's coated with collagen, and then you see the cells, and initially they're grown submerged, just like you would grow any cell line. So there would be media on the top and media on the bottom. But then after a couple of days of growth, allow the cells to spread across the well, you aspirate the media off the top, and you only feed the cells from the bottom, and that allows these cells to polarize and form cilia. And so they look a lot like native epithelium. And in fact, these are the types of cultures that were used to develop the new drugs that we have for cystic fibrosis, that work on correcting mutations that result typically in misfolded cystic fibrosis transmembrane regulatory protein. And so, but they've also been used for host defense studies. And so what they did was they did in situ hybridization for ACE2 in the nose and in TRM2. And you can see that they're both expressed here in these nasal cultures. And in fact, they're higher in the nasal cultures than they are in the trachea cultures, and even higher in the alveolar cultures. But there is data that ACE2 can be induced. So particularly in response to viral infection. So one potential is that, so they argue that there was a gradient actually with the highest ACE2 in the nose. And then, so this may allow the virus to at least get started in the nose. And if some of the virus is aspirated down in the lung, there's some ACE2 expression that allows the virus to start inducing an interferon response. And then these interferon responses can actually induce ACE2 in the distal lung. And then, work by Chad Roy at the Primate Center has also shown that, another model of infection would be very small aerosol transmission, that it could even be so small that that would bypass the filtering of the nose. The nose generally filters, particles above 10 microns. So there could actually be direct inoculation of the lower airway, and this is clearly supported by the fact that masks reduce disease and blocking this. So, so what about the host genetics? So, so of course, not everybody that gets COVID or gets infected develops severe COVID. So this was this severe COVID-19 GWAS group. This was a European study, and they published in the New England Journal. And what they found was blood group antigens are an important covariate here. And then, but interesting, there's a number of genes that clustered on chromosome 3 here. And if you notice, there's 3 chemokine receptors. So there's CXCR6, XCR1, CCR9. And then these other genes are actually, these 2 genes, LCTFL1 and FYCO1, are expressed in dendritic cells, which are one of the antigen-presenting cells in the lung. So for CCR9, this is a gut-homing T-cell receptor. So it's a little bit unclear how this would regulate, pulmonary immunity to virus, because we think of CCR9 as pretty much the gut-homing T-cell receptor. So, for example, pyre patch formation in the intestine. But XCR1 is a chemokine receptor on NK cells, natural killer cells, which can kill viral infected cells, just like they can kill cancer cells. And then CXCR6 is a marker of what we call tissue resonant memory cells, or what we call TRM cells. And I'll discuss these cells a little bit more. So TRM cells have been defined over the last decade or so by studies largely led by several labs, but I would say Donna Farber has done the most. So Donna is at Columbia in New York, and starting about 8 or 10 years ago, she got together with all the transplant groups in New York City, and any lung that was destined to be transplanted, but then for some reason couldn't be transplanted because of maybe the lung was contused or some mismatch or size mismatch, and so the lung was going to essentially be discarded. If the donor family had consented for research, she got the lung into the lab. So she studied over 400 lungs over this period of time. And what she's found, very similar to what's in skin and the GI tract, are these tissue resident memory T cells that are only in the tissue. They don't circulate in blood, and they don't home back to draining lymph nodes. And the reason why is because they don't have these egress cues. So they lack CD62L, which is important in trafficking to lymph nodes by interacting with the high endothelial venule going into the lymph node. S1PR1 is a G-coupled protein receptor that's involved in egress, as well as CXCR1. These are all GPCRs. And they're thought to be what maintains or at least keeps the cell from trafficking back to the lymph node. And then they have these adhesion molecules involved in tissue retention, one called SIRTAM. This is actually what keeps your Peyer's patch cells in the gut, essentially. And then about 50% are CD103 positive, and about another 50% are CD103 negative. And they seem to be in an activated state, so they express this activation marker CD69. And then they come in all types of flavors of T cells, anti-inflammatory cells that make IL-10, and then effector cytokines like interferon gamma or IL-17. So it's thought that these cells are induced by antigen, inhaled antigen over time, and their memory, so they can recall prior exposures. And these are the cells that may that can activate rapidly if you see an autologous antigen. So, for example, if you had a prior coronavirus exposure, an alpha coronavirus, but there was some shared homology with that alpha coronavirus with SARS-CoV-2, particularly in the structural antigens, that these cells actually may provide some protection, and this is why they may have showed up in that GWAS study. So the SNP that was identified in the GWAS study was a SNP that actually reduces CXCR6 expression, so less homing to the tissue site. So I think it's an interesting hypothesis and this could influence how we want to vaccinate, because the current mRNA vaccines right now are largely going to generate a systemic antibody response that will neutralize the virus, but they're not going to generate this type of T cell response. The only way that you can really generate this T cell response, at least easily, is with a mucosal route of immunization and not an intramuscular or subcutaneous route of immunization. So, so I think novel vaccine approaches could be exploited to try to target these cells, particularly since we now know that they're part of the natural immune system of the developing lung. It's interesting that in the infant lungs that Dr. Farber has studied, is that most of these cells are what we call T regulatory cells, or anti-inflammatory cells. And so this may be an important mechanism of why not only 5% of children develop asthma. We know that there's an active tolerogenic mechanism to not respond to every inhaled allergen or antigen, and so these cells may be playing an important role in also limiting responses or regulating responses in the lung. So this is some data that we collaborate with Juan Celadon's group at Pittsburgh, and Juan has been studying childhood asthma, and as part of that, he's been looking at RNAseq in the nose, and this is actually single cell RNAseq, and he wanted to know. He's looking at whether patients with asthma have a different single cell profile in nasal brushes compared to patients without asthma, and so we collaborated with Juan and said, are there TRM cells in the nasal mucosa of healthy children? And indeed there are. So you can see here's the, they actually published this data and they just called this the immune cell cluster. But if you drill down and look at the immune cell cluster, there are CD8 positive cells, there's CXCR6 positive, and then another marker that we like for TRM cells is ICOS positive. So the question then is, what's the antigen specificity of these cells? Are these, this is pre-COVID, so are these rhinovirus specific, or what's driving these cells? And can this explain the reason why children get nasal infection but don't progress to COVID-19 for the most part? So, so we recently, James Robinson's group got a U54 grant to look at serological responses in blood, but we're hoping to do nasal sampling on a subgroup of children that have COVID or had SARS-CoV-2 positivity infected, and then see does the nasal infection result in the generation of viral specific CD8 cells in the nose that may protect them from future infection. So I'm going to show a couple preclinical vaccines. Obviously, we got EUA approval for the Pfizer vaccine last week, and I haven't heard yet today whether, but it sounds like the Moderna one's going to be probably approved today. So the question is, should we consider mucosal routes in addition to the systemic routes, and do they offer any advantage? And the first group that published a mucosal immunization that I'm aware of is Mike Diamond and David Curiel's group at Washington University in St. Louis. And here they compared intramuscular immunization with this is an adenovirus vaccine, so very similar to the Oxford vaccine. This is a chimpanzee adenovirus encoding the spike protein of SARS-CoV-2. And then so they did a prime, then a boost, and then in other experiments they blocked the interferon receptor to see if that blocked. They used this to try to make the mice more susceptible to it. And then to test SARS-CoV-2 infection, so mice aren't susceptible to SARS-CoV-2. So to make them susceptible, they had to give an adenovirus encoding human ACE2 down the airway. And that now puts human ACE2 on the lung epithelial cells, and now the mouse becomes susceptible to SARS-CoV-2 inoculation. So on unvaccinated mice, you can find viral replication and high lung titers, but in the mice that got the vaccine, essentially no negligible viral growth in the lung. This is by plaque formation. They also looked at lung viral RNA using real-time PCR, and you can see that there's a significant reduction at day 4 and day 8. And then another biomarker of pulmonary infection is also the virus induces chemokines associated with interferon signaling, such as CXCL10, and there was a reduction in CXCL10. But what's interesting is that they got no mucosal IgA response. They only got a systemic IgG response with this vaccine. And so you can see that there's some viral RNA here. So it's a pretty dramatic response, but not, I wouldn't call this sterilizing immunity because you still have some lung viral RNA here. So then in the, when they gave the vaccine intranasally, they were able to elicit a local IgG response in what's called the BAL, that's the bronchoalveolar lavage fluid or the lung lavage fluid, as well as an IgA response. These had a high neutralizing serum titer and serum neutralizing titer. And then here, there was basically no detectable virus 4 days after the inoculation. So this suggests that the mucosal route was actually more robust, both in terms of plaque reduction as well as in viral RNA. And actually, importantly, it protected viral replication in the nose. And I don't think the current vaccines were going to do a very good job of that because systemic IgG just doesn't have great penetration into the nasal airway. So this is a model, and what they don't know is what's the relative, how is this IgA response generated in the nose with this type of vaccine? Is it antigen presentation by dendritic cells? Or, since this is a big particle, and adenovirus is about 100 nanometers, the larger the antigen, potentially the more important role B cells play in antigen presenting cells. So the respective role of dendritic cells versus B cells and antigen presentation in this, I don't think it's yet to be determined. And then, do you need trafficking back to the lymph node? The lung's a little bit unique. So the lung has lymph nodes, so you can get trafficking back to the lymph nodes to generate this T follicular helper response and germinal center reaction, but the lung can also form what's called eyeball, tertiary lymphoid structures that are actually formed just below the airway. And therefore, you don't need trafficking all the way back to the lymph node to generate a mucosal response. And so it's a little bit unclear, this pathway. So, but interestingly, this vaccine this is based, the reason why they use the chimpanzee adenovirus is that there's no pre-existing immunity in humans. The trouble with other adenovirus serotypes is like we have pre-existing antibodies to like Ad5, for example. So there's concern that they won't be very effective. But anyway, this group has licensed this vaccine to a company in Gaithersburg called Altimmune, and they're very interested in intranasal vaccines, not only for SARS-CoV-2, but influenza as well. So I think we're going to see more and more companies try to target the nasal mucosa for vaccines. And then this is the issue of cross-reactive T cells. So this is data from Mark Lipsitch and Shane Crotty's group, and they've looked at some of this T cell cross-reactivity, and their model here is with COVID. If you have no cross-reactive memory, then you get this huge viral replication, and then you got to generate both an antibody response here in purple, as well as the T cell response in blue here. And if you're very efficient in doing that, you may limit severity. But if you're not, if you're totally naive, then this may be the patients that develop the most severe disease. But if you have some cross-reactive T cells, then you can actually reduce the viral load, because your T cells are going to be coming up much faster if you have memory cells than if you're completely naive to the and if you have tissue resident memory cells in the upper respiratory tract, then you may limit disease altogether. So this is kind of just a schematic representation of the model that that a lot of people are thinking of how this may work. And and if this is true, again, this supports the idea of developing mucosal vaccines that target this population, in addition to systemic antibody. So let me just touch about animal models that the pharma and academia is used for modeling COVID-19. So I mentioned the adenovirus human ACE2 model. There's also the K18 transgenic ACE2 model that Paul McCrae generated in Iowa. He's a pediatric pulmonologist there. Work with Stan Perlman. And this cytokeratin 18 is just an epithelial promoter, and it drives ACE2 in many epithelial tissues. And this was actually developed in response to the original SARS outbreak in 2004, and or actually in 2002/3. So this, interestingly, when you inoculate this animal with SARS-CoV-1, the original SARS coronavirus, they develop a lot of lung disease, a lot of viral replication, but they also develop dissemination of the virus to the brain, because this K18 is expressed in the ependymal lining of the brain. So these mice actually die of an encephalitis. We see mortality with SARS-CoV-2, and we're still Zubin Chen's group at the Tulane Primate Center, who's really developed this model here at Tulane, is still trying to discriminate between how much mortality is CNS infection versus pulmonary infection. But the bottom line is, if you give enough SARS-CoV-2, 2 times 10 to the 5th, PFU, this is a lethal model. A number of labs have tried to make human ACE2 mice. This is knocking human ACE2 into the endogenous mouse ACE2 locus, so it's controlled not by an ectopic transgene like K18 is, but it's controlled by the native locus of the ACE2 gene. And then Ralph Barrick's group has made a mouse-adapted SARS-CoV-2 virus by basically just passaging human SARS-CoV-2 serially through mice until you get one that can replicate in mouse cells. And then there's natural models. The hamster seems to be susceptible, and a lot of groups are using the hamster model. Ferrets also seem to be susceptible, guinea pig, and of course the non-human primate model that several groups have published, but Jay Rappaport's group at the Primate Center has recently published data from both rhesus and African green monkeys that they can support some pathology after SARS-CoV-2 inoculation. So what about human models? So as I mentioned, we have these air-liquid interface cultures that can be also used to model SARS-CoV-2 pathogenesis. And then you can also now take alveolar cells and form organoid cultures, which also can model the more distal lung, the type 1 cell and the type 2 cell. So we've collaborated with Barry Stripp's group at Cedars-Sinai, who's developed these, and you can see that these are air-liquid interface cultures where you infect with SARS-CoV-2. And interestingly, most of the cells that are at least infected initially are ciliated cells. So these acetylated, this is acetylated tubulin and marker or ciliated cells, and you can see the virus really co-localizes pretty well with these ciliated tubulin cells. This is MUC5AC here, which is a goblet cell marker, and you can see that there's very little infection. There's some infection, but a lot of the goblet cells remain uninfected in this model. But we don't really know how this virus spreads. So once you get an infected cell, does it spread contiguously, or can, does it spread through cell death and release of virulent particles? I think that's an important area that needs to be investigated. And then these are the organoid models, and you can see that they grow in spheres, and you can see there's pretty broad infection in these organoids, including both type 2 and type 1 cells. And so these models, if you treat with interferon beta, you can reduce the number of infected cells in this model. There was a small effect of hydroxychloroquine in the large airway model, but no effect in distal organoids. And then remdesivir, given prior to viral infection, reduces viral load in these, both in the upper airway and the lower airway organoid culture. So these in vitro models can be used to test antiviral compounds. So I mentioned the adenovirus 5 ACE2 model. This is work from Zuben Chen and my lab here at Tulane. So we have an ABSL3 facility on the 5th floor of the JBJ, and this again, we just transduced mice with an Ad5 ACE2 inoculated with SARS-CoV-2, and you can see that there's productive viral infection in these, and you don't see this at all in mice that get a control adenovirus. This viral replication is clearly dependent on the expression of human ACE2. And here's the ACE2 levels by real-time PCR. It's fairly, it peaks on day 4, but you still get substantial expression of ACE2 in the whole lung out to day 14. We did not see any viral infection in the intestine because there's no ACE2 here. So if you give the adenovirus, does not transduce the intestine in this model. And then this is immunofluorescence showing, this is actually a SARS-CoV-1 antibody that cross-reacts with the nuclear protein of SARS-CoV-2, and you can see viral infected cells. And there's a number of, this is all distal lung, so this is all type 1 and type 2 cells. And then we did RNAseq, both for viral RNA, but also for the chemokine CXCL9 and 10, and you can detect high levels of CXCL9 and 10 induced in this model. So we like RNAseq because it's an unbiased assessment of what the lung's doing. You can see that in this volcano plot, there's high induction of interferon gamma, CXCL9, CXCL10, IL-21, which is important in both T cell immunity, but also in generating class switch immunoglobulin. It signals to IL-21 receptor on B cells is induced in this model. And many of these genes that we saw induced in the mouse model was shared in infected human lung at autopsy. Now, there's obviously a lot of genes here that are different, almost 1,000 genes up in the mice. But this is obviously day 3 in the infection, and this is end stage. But what was interesting, we saw induction of many chemokines, including CXCL10, again, this interferon gene, MX1, which is another antiviral gene, and then the C3 complement receptor. And actually, the fact that antibodies are associated with more severe disease, one wonders whether the complement pathway is really important in SARS-CoV-2 pathogenesis. So I'm just going to end on, our fledgling efforts to try to develop some therapy particularly for patients that may not be good vaccine candidates, like transplant patients. And it was very clear very early on that this virus used ACE2. Second, this virus had higher affinity to ACE2 than SARS-CoV-1. So therefore, there was some data that maybe even had picomolar affinity. So if it had picomolar affinity, well, that's in the druggable range, right? So like a lot of monoclonal antibodies have picomolar affinity for the target. And this is Enbrel. So Enbrel is TNF receptor antagonist. It's the P75 TNF receptor fused to an IgG1. And the reason why this drug works is because you can see here's a schematic. This is a hidden Markov model of the TNF receptor. And what we're looking at here is the pink is the extracellular domain, the red is the transmembrane domain, and the blue is the cytoplasmic domain. So TNF receptor family, just like the nerve growth receptor family, these are single pass receptors through the membrane. Well, so is ACE2. So you can see that there's this short little cytoplasmic domain, the transmembrane domain, and then, so this is what we call a type 1 transmembrane protein, where the N terminus is on the outside of the cell and the carboxy terminus is on the inside of the cell, and it's a single pass. So this means that soluble ACE2 can be engineered into an IgG fusion protein, probably just like Enbrel was. So that's what we did. We cloned this and fused it to human IgG1. So this is kind of what this molecule would look like theoretically. We retain the disulfide bonds here, and then the human ACE2s are here, and then IgG1's here. And then we tested to see whether this could neutralize virus both in vitro and in vivo. And then we made a couple modifications to the protein. So first, we mutated the FC to abrogate FC receptor binding. So there's a well-known mutation called the LALA mutation that you can introduce into IgG1 that will abrogate FC binding. And the reason for that was, particularly early on in COVID, there was this concern about cytokine storm, and we didn't want to activate FC receptors. The second thing is that for this to neutralize, I don't know if you noticed that Donald Trump got 8 grams of the Regeneron antibody. So you got to give high doses of this. And if this had functional ACE2, that's a lot of biologically active enzymatic ACE2 floating around, and who knows what that would do to somebody's blood pressure. So we mutated the catalytic domain of ACE2 to avoid any hemodynamic side effects. And knowing that the catalytic domain of ACE2 was actually very far from where ACE2 actually interacts with SARS-CoV-2. So first we did binding protein studies, so just to see, does this molecule bind spike? So here's wild type ACE2, and actually one of the mutants we made, which we called MDR504, because 504 area code for New Orleans, bound the spike protein with higher affinity than wild type. And this is we verified this is catalytically dead. So this is a fluorogenic substrate that mimics ACE2 catalysis, and you can see that the wild type protein catalyzes this fluorogenic peptide and releases a fluorescence signal, whereas this molecule does not. So then we did a couple of viral neutralization assays. So at this time, our BSL-3 wasn't up and running, so we sent protein to Ralph Tripp's lab at University of Georgia, world-class virologist, and Ralph was kind enough to run these in a plaque assay. So this is viral neutralization. So the IC50 here of our molecule MDR504 is about 0.6 nanomole, which is pretty good. There are some more potent monoclonal antibodies, but the risk of that of a picomolar monoclonal antibody is it's that epitope is probably, if there's a single amino acid mutation in that epitope, you could get easy escape resistance. So I don't know if you want. That's the downside of that. And that's why Regeneron actually has a cocktail. They're giving more than one antibody to precisely try to prevent escape resistance. So the nice thing about an IgG1 fusion protein, just like a monoclonal antibody, this is serum pharmacokinetics. It's got a very stable half-life, approximately 114 hours in serum. And since it's an IgG1s cross the placenta, but they also cross into the lung through the neonatal FC receptor. And you can see that we get very high levels in the bronchoalveolar lavage fluid. And so the idea of this molecule is to really prevent the distal lung, the type 1 and type 2 cells from getting infected. So if we could treat early, that we get high enough concentrations to prevent that from happening. So this is a prophylaxis model here. And what we're looking at is day 4 after infection, where we've treated mice with vehicle control, or this ACE2 FC that we made, and just the number of green cells is the number of infected cells. And you can see that there's a significant reduction of infected cells when it's given prophylactically. And when you block infection, you also block induction of CXL10. There's no interferon response because there's basically just no virus. And this is just confirmed with this RNAseq, where CXL9 was significantly reduced in the treated mice, particularly compared to wild type ACE2. This mutant seems to be superior. So then what about a treatment model? So here we moved to the K18 model, and we gave it 4 hours after viral inoculation. This mouse supports very high viral titers. So you can see 10 to the 6th, 10 to the 7th, and we were able to significantly reduce viral load at day 3. And we've gone on to do one survival experiment where there was a significant trend for that there was really no survival in the treatment group and 50% mortality in the control no treatment group. So there appears to be a survival benefit of a single dose of this drug. So these data went into an FDA IND last week, and we're waiting to get approval from the FDA to conduct a first-in-human phase 1 study. So the and then again, this shows that it reduces CXL9 expression. So I want to thank everybody that's done the work here. Naoki Iranaga did, and I designed the protein, and he produced it in CHO cells. And then we did the mouse modeling work with Zubin Chen's lab and Kun Han, and then the Tulane ABSL3 core, which is led by Christopher Mandra on the technical side, and then Angela Birnbaum on the IBC biosafety side. And then Ralph Tripp's lab did the neutralization assays for us, and then the wrong lab at UIC, led by Yuzhong Rong, he helped us with a pseudovirus assay, and this is some of the lab people. So I'd be happy to answer any questions. So I ran over, and I thought I could get it done sooner. Thanks.
Jay, there was a question from John Kerwin. He said, great overview, thank you. Could you comment on the likelihood that we will see multiple variants of the virus? There appears to be a variant that has cropped up in the UK.
Yeah, so there have been mutations in the spike protein, even, and I get, so we donated our protein to what's called COVID. It's the Gates-funded operation out of Loyola Immunology Institute. And so they collect every monoclonal antibody that people have made. And as soon as a variant pops up, they test it. So they've already tested our protein to one of the spike variants, and actually our protein actually bound better with the mutant. So, I think the advantage of the vaccine is it's going to generate a polyclonal antibody response. So it's going to put multiple immune pressure on the virus, and probably, hopefully, ideally, maybe if enough people get vaccinated, we can eradicate the darn thing. The trouble with monoclonal antibodies is they're going to bind at really distinct epitopes, and so escape resistance is definitely more of a concern with the monoclonal antibodies. But it's an RNA virus; it's going to mutate, but I don't think it's going to mutate enough to make the whole protein-based vaccines basically. I don't think it'll affect their efficacy. It's not like influenza, which is a segmented genome and can reassort, and it has influenza has eight segments, and the reason why it's such a risk is that it can reassort in addition to mutate.
All right. So Megan, what do you think?
Good question. We're really close to our ending time, everybody. I hate to ask Dr. Bojanowski to reschedule, but we don't really have enough time left to do your talk justice. Is that true? Okay, so thank you, Dr. Kolls, for your amazing talk.
No problem.
Unfortunately, we run out of time. So.
Yeah, no, I think that's fine. And so just to everyone, of course, thank you to Dr. Kolls for that great talk, and to all of the attendees today. A recording of today's session will be available in the next 1 or 2 days. It's going to be shared on the LaCATS listserv and posted to the LaCATS YouTube channel. The next LCATS COVID-19 Chronicle Seminar will be held on Thursday, January 14th at 4 p.m. At that time, Dr. Paul Fidel, who is with the LSU School of Dentistry, and Dr. David Welsh from LSU NO will be presenting. We welcome more speakers for the seminar series. So if you or someone you know would like to share their COVID-19 related work, please email info@lacats.org.
All right. Thank you so much, Dr. Bojanowski. You did a phenomenal job hosting, and we will do our best to find an alternate date for you to present your works, because there's always going to be new trials to talk about. So I'm sure in the future there'll be that many more slides that you can add. So thank you so much for being here and for joining us today. It was great to see so many attendees. If there's any additional questions, feel free to pop them in the chat real quick. We can take, you know, the next 9 minutes to answer questions if there are any. If not, we'll give you back you guys' time. Have a lovely rest of your Thursday evening. And thank you, Dr. Kolls. This is really enlightening. There's a lot of tremendous work being done, and it's exciting to see it being done so quickly. This is amazing to me.
Yeah. Sorry, Christine, I should time myself.
They're buzzed. Burn. No, that was a great talk. So.
Okay. Thank you all.
All right. Thanks.
Have a good holiday, okay?
Absolutely. Happy holidays, Dr. Kolls. Happy holidays, everyone.